So the overview will be, I'll talk really sort of briefly about drug discovery and drug development. I'll also give a high-level view of large language models, and then I'll go just a little deeper into BERT, which is a specific type of large language model. And then I'll go a little more detailed into models that predict things about proteins. So this is useful for drug discovery. And then I'll talk a little bit about how I train these models. I'll talk a little bit about the hyperparameters. Some of them are kind of tricky. And then, and as I said, questions along the way are very, very welcome. So don't hesitate to pipe up. So, the goal, of course, of developing drugs is to develop safe and effective drugs. And by safe and effective, we mean things like you want this drug to be as potent as possible, and you want it to have as few side effects as possible. And as we saw with COVID, we always need to be aware of being able to treat new variants as some of these viruses will change their characteristics. So a drug that works now might not work in about a month or so, or however much time. For COVID, it was like about every, I think, 6 months. There was another significant variant. So there's a vast, vast number of potential drugs that someone could try to develop for various viruses or infectious diseases. And there's a vast number of drug characteristics that make a drug a quote unquote good drug to market. And so some of those characteristics may be things like, how potent is it? How quickly does it metabolize in the body? What kind of shelf life does it have? When a chemist develops the drug, what's the yield when this person is creating the drug? So for example, or does this drug need to be refrigerated? So you might imagine that if a drug needs to be refrigerated, it has a short shelf life. You always have to have refrigeration in order to dispense the drug, and that could be problematic. So there are all these characteristics that if we could fine tune them and make it so that this drug is more potent, safer, and easier to manufacture and distribute, then that's a big win. But it turns out that trying to figure out, what drugs are good and then, what drugs are better than good is a really slow and costly process. I believe the last I heard, it costs something like millions, $2 million, $3 million or maybe even $10 million to develop a drug. So it's really quite expensive. So one of the things that I was involved with in my previous job was, instead of having a person manually do this process of tweaking a drug, and I'll go a little more in detail about what tweaking a drug means. What we tried to do in the machine learning group was do what we think of as virtual screening, or in silico, meaning programmatically. And so not that my models are gonna discover a new drug. Instead, what we were aiming for is, because there's such a large number of potential drugs, we were trying to narrow that down so that our chemists and our druggists didn't have to waste time on drugs that we knew were going to be a complete failure. I was also trying to identify, bad characteristics of drugs and saying things like, let's not go down this avenue because this makes the drug more toxic, say. And we were hoping our models would be able to identify drugs that may be less effective. So again, so that a chemist doesn't have to waste their time trying to, screen through them. And when I talk about screening, so this is an example. This is actually some of my data, and I'll try to orient you to this. This is called a mutation map, or a deep mutational scan. And essentially on the x-axis, you see a bunch of little letters. This is the representation of E. coli. Each one of these letters is an amino acid. So the first one is methionine. The second one is proline, that P. The next one, R, is arginine, and serine, and cysteine, and cysteine. So each one of these is an amino acid in this drug. And then if you tweak that M, that methionine, and make it an A, and I don't know what A is actually. I'm not even sure it's a thing. But if I've changed that one amino acid from methionine to this A, what happens? Is the drug more potent? Is it more toxic? Who knows? But because there are 26 amino acids, for every one of these amino acids along the x-axis, if I change it and mutate it to a different amino acid, A, B, C, D, E, all the way up to Z, what is now this drug's characteristics? Like, for example, again, let's say I'm trying to measure binding strength, and maybe we want the binding strength to be as strong as possible. So if I change this first amino acid from an M to an A, is it more or less strong in the binding characteristic? And so this is always compared to, again, this is E. coli. So this data, the string down here, the amino acid in the bottom, this is made up. I mean, not made up, it's E. coli, but this data is not for E. coli. I kind of wiped it out so you can't see because it's kind of proprietary. But so these are the sorts of maps that I'm trying to create with my models, which my models say, oh, okay, if I take E. coli and change one of these amino acids from M to A, it predicts what is the potential, it predicts, it doesn't know for sure, but it predicts this characteristic. So hopefully that kind of makes some sense. I will assume that means yes, since I cannot see you. So that's kind of a very high level overview of drug discovery and drug development and where these large language models come into play. So now I'll just talk very high level about large language models. They are a type of deep learning model. They're a neural network, and they're an iterative network. And by iterative, what they do is, and for those of you who have done modeling before, you've probably seen this before, but essentially the model will go through its paces and make a prediction. And once it has that prediction, it will compare it to truth. And then when it's all done with that, it says, oh, okay, some of these predictions I got right, some of these I got wrong. Let me go all the way back to the beginning and change some of the neural nets or some of the weights in the neural net and make another prediction. And the goal is to get more and more correct. So it does that over and over and over again until the machine learner program data scientist says, all right, that's good enough. You don't have to train anymore. So these large language models that we've been hearing about quite a lot recently, they're trained on natural language text, and they're trained on things like Wikipedia pages, reviews in Yelp, IMDb, SQUAD, and SQUAD stands for Stanford Question Answering Data Set. And so that's what goes in. The input is a bunch of words, and the output is supposed to be a bunch of natural language text. And the goal is for the text to be correct based on the prompt, the input, and also to be fluent, to sound like what you would imagine a human would say. So some applications that we've seen for large language models are language translation. So for example, there's a ton of text out there in English, but there may be some languages that aren't really well represented on the internet, and you'd want to translate those things. So rather than starting from scratch, you can use this large language model and tweak it a bit so that maybe it'll translate to, I don't know, some dialect that there isn't a ton of text out there. It can do text summarization, which is really nice for me. Sometimes I'm reading papers and I don't feel like reading them, so I ask for a summarization, although that's what an abstract is for, but whatever. But it could also do sentiment analysis, which is nice for marketing. Does someone like my product or not? Do they like my restaurant or not? And then there's chatbots, and you can, for question and answering. So for some examples, a lot of people have heard about ChatGPT, which was released by OpenAI. And the new Bing from Microsoft is built on ChatGPT. Google has also released Bard, and Meta, Facebook, has released Llama. So these are some examples of large language models. So before I get into some sort of conceptual explanation of how what these large language models are doing on a high level. Are there any questions? Okay, all righty.
That has been quiet so far, so hopefully you'll start adding there. Yes.
Okay. So this is the way I think of large language models. I think of them as a massive, when you're texting on your phone, it'll make suggestions of what the next word should be. What is that called? Autocomplete. So I think of large language models as being massive autocomplete tools. I'm sure there are people who would disagree with me, but at least conceptually, this is what works for me. And so I'm just going to try to describe in that framework what a large language model does. And so, or you can also think of it when you're typing at like in a web search engine, if you start typing things and it wants to try to complete what it thinks you're going to type. So what it does is when it sees you type something, I saw a cat, it's going to try to give you the next word. And so here's some examples of potential next word. I saw a cat on the chair. I saw a cat running after a dog. So there are many, many combinations. So, how does it do a good job, actually? The other thing I'll mention is this website by Lena Voigt. She's part of Meta Facebook AI. This is actually a really good webpage, or a nice explanation about what's going on. She's got lots of pictures, and that's what these are from. Almost all these images are from her webpage, and so I highly recommend it. So this is what a neural net is doing, is it sees me input, which we're calling a prompt. It can see me input, prompt it with the word I. And so it says, okay, given that you've given me the word I, let me go look at all the words that I know about and see what other words typically follow the word I. So basically it tries to find the highest probability of all the words that are next to, if you will, the word I. And so that's what these gray bars are saying. Each one of these horizontal gray bars is a word. I what? I saw, I went, I swam, I ran. And each one of those words has a probability of how likely it is to follow the word I. And so a massive auto complete will say, okay, I ran, or whatever. And then you all of a sudden type saw. And so it's like, okay, now you gave me two words. You typed I and saw. So let me get all the words that have the highest probability, and I'm going to rank them in order of probability of the words that typically follow the phrase I saw. And then you type A. So now the larger language model is, says, okay, you typed I saw a. What are let me rank all the words in highest probability to lowest probability order. What would be the next word that follows I saw a? And so it goes on and on and on. And we all know the internet is a huge database for cat pictures. So it's not surprising that maybe the next word is I saw a cat. And then if I typed I saw a cat on, the large language model is going to try to come up with the next word. So in this case, if I'm typing I saw a cat on a, it might in the end give me Matt. So in the end, what a large language model is doing is it's trying to find the probability of the next word. And it's always trying to make that next word had the highest probability, but there are a couple of knobs that can be tweaked to make it not always be the highest probability. But that is essentially what's going on in these large language models, is it's looking for high probabilities of words, given that you've already given it some words. And so these models are called left to right models. And they're called left to right models because it's always looking at, well, at least in the English language, we read from left to right. So it's as if the model is reading from the beginning of the sentence on the left and marching to the right with each word you give it. Does that make sense? Okay. So now what I'll do is I'll just describe a little bit like the anatomy of these large language models. And so you can see here on on the bottom here, I saw a cat. That's what I'm inputting, that other people call a prompt. I'm inputting, I saw a cat on And then this middle purple area is a neural network that is working on this input, and it produces some output. Now, it turns out in this simple example, this is a neural network with one layer, but there are neural networks or large language models with many, many layers. And so what's interesting to me about these many layers is it's really cool that these layers seem to naturally order themselves from the earliest layers. Layer 1 is typically the simplest layer. And then as each layer continues, it gets more and more complex. So in the case of language models, people can see that the earlier layers are sort of recognizing the syntax of, in this case, English. It's understanding parts of speech. It's starting to recognize named entity recognitions. And then later layers seem to start to understand semantics. It starts to understand context and grammar. So this concept to me is super cool. And so in order to try to make this clearer, I want to talk about, I'm going to switch for just for a minute and stop talking about language models and start talking about image models. And the reason I'm doing that is because I can show you images that have been published in papers, and this is easier for me to understand. So I'm hoping it'll be easier for you to understand. So here in this upper left-hand corner is the first layer of an image neural net. And it's starting to see patterns here. And these patterns, like this pattern in the upper left-hand corner of this box, it's recognizing these pieces down here. Hopefully you can see. Let me see if I can change my pointer, laser pointer. Okay, I don't know if that makes it easier to see. But this corner up here is recognizing these pieces. And so here are sort of like diagonal lines. And down here, the lower right-hand corner, it's seeing things like gradients from red to green. And the lower left-hand corner, it's seeing like patterns that are made up of dots. Okay, you know, that doesn't look like recognizable of anything in particular to me, but it's like a piece. And so the second layer gets a little more sophisticated. And so again, this upper left-hand corner is identifying these images in the upper left-hand corner on the right. So what I find fascinating is, look at the lower right-hand corner. You see these sort of right angle pieces. It's recognizing edges of like doors and windows. So now instead of just seeing like single lines, now it's seeing 2 lines. And then it starts to see circles. And now it's over here, it sees half circles. So you can see it goes from something very abstract into something a little less abstract. Now we have circles and corners. And then if we look at, I just skipped a layer, I skipped layer 3, but if I look at layer 4, now check this out in the upper left-hand corner, that is a dog face. And nobody said, hey, this is a dog face. It kind of self-organized that way. Here's dog faces. Down here, these are legs of birds. They're not just any legs, they're legs of birds. And if you look at layer 5, this is fascinating to me, this lower right one. These little circles, they're not just circles, they're eyes. And they're not just anybody's eyes. They're eyes for birds and reptiles. They're not just circles. And if you look to the left of that, here are more circles, but they're not just any old circle, they're wheels. So you can start, at least I can, start to see that if we go from the first layer where it's just kind of, you know, a basic building block, all the way up to this 5th layer, although layer 4 with these dog faces is pretty awesome to me. I mean, that's pretty cool. So now going back to, so before I go back to large language models, just want to check, anybody have any questions? Hopefully you're super excited about that, because I am. I think that is the coolest thing. Do you think that's cool?
Yeah, that's pretty interesting.
Cool, excellent. Thanks for the input.
No, I work with imaging of the brain, images, and I know that was like 13 years ago, that it was really difficult to do sharp edges, to even like not even that sharp, but like 70-degree angles were very difficult to resolve. So this is incredibly cool. Yeah.
Yeah.
Yes, you're
So,
getting similar on chat. Very cool. It's amazing and starry eyes. So everyone's with you, I believe.
awesome, awesome, great, great. So now, so I'm gonna put aside the image, large models, because I was only doing that just to try to explain something. So now we're gonna return to language models. This is the same picture as I've been showing you before, but I tilted it 90 degrees on its side. So again, input from the left instead of from the bottom, the layers of the neural net in the middle, and then there's an output. So let's just talk a little bit about some vocabulary that is often used when talking about large language models. These layers are typically called a body, and it's called the pre-trained part of the model, and the fact that someone has fed massive amounts of input through this neural net and let it iterate over and over and over, sometimes for days, on massive amounts of compute and memory resources. And so the result of that is often called the pre-trained body. And so what I find incredibly fascinating, again, is that people will develop these massive bodies and put them out in Hugging Face. They just put them out there. It's like a repo. That's the best way I think of it. And they're free. They just put them out there for free. So for those of you who haven't interacted with Hugging Face, I totally recommend you can look at this models tab, and if you can type a couple of words and see, this is how I did my work. I went to models and I typed proteins, and I found a bunch of models that someone trained and just put out here for me to grab. Because what this means is I don't have to do this massive amount of training. And what's really awesome about that is I don't have the amount of data, and I didn't have the resources. I never would have been able to do this. So I just think it is amazing that people are this generous. And I think Hugging Face is a great place. Okay, so back to this. All right, so far we know about the body. And so suppose now this large language model is trained on a bunch of English words. And suppose I want to translate to French. So what I can do is put my own layer on, and that layer is called a head. It's the head layer. And this head layer is fed my English to French lookup table, if you will. And so what I do is I take the output that was built on English and I fine tune it on French. And then instead of outputting words in English, it'll output words in French. And so again, what is amazing about this is all this stuff back here in the body, I don't have to touch that. I don't have to train that if I don't wish. I can just do this fine tuning of one layer, this head layer, and that can be as quick as 15 minutes. So I can basically build on top of this large language model that someone took days, if not weeks, to train. And so these kinds of models are called transformer models, and it's part of what's called transfer learning. Again, in the fact that someone's already done all the heavy learning, and I just tweak it, fine tune it, so I can transfer the learning from the body over into my space, translating English to French. Okay. So now that we've talked about large language models at a high level, I want to talk again in a high level about a specific large language model called BERT. And so BERT is a specific type of large language model, and BERT stands for bidirectional encoder representations from transformers. So there's that word transformer again. So it is a large language model, it is a transformer. So what makes BERT it really got a lot of people's attentions about its accuracy. And one of the strengths of BERT is that it's bidirectional, meaning you may remember I talked about that autocorrect model, that it was only left to right. Well, bidirectional for BERT means it can look at context before and after a word. So if I gave you this sentence, we went to the blank. Well, blank, that could be anything. We went to the circus for all I know. But if I gave you a sentence, we went to the blank to make a deposit, you might be able to say, oh, okay, you went to a bank. So BERT can look at words afterwards as well. And so that made it pretty powerful. The other nice thing about BERT, it's not just a large language model, but it's also a masked language model. So again, remember the training data is a lot of text from Wikipedia, I think Reddit as well, but lots and lots of text. And so the way it got trained is, let's just say you give it one Wikipedia page. In the beginning of the training, it'll take that one page and it'll blank out 15% of the words. And then it'll feed that through the model. And the model is supposed to guess or predict those missing words, the 15% missing words. And when it's done, it says, all right, I know what the right answer is because I masked them out. And so again, it does what a model typically does, is it says, all right, I predicted these things. Let me compare them to truth, to the correct answer. I got some right, I got some wrong. Let me start all over again. Let me iterate and let me go through this neural network and all these layers and tweak them to see if my next prediction can be better. And it does that over and over and over again because I've masked out 15% of the words. The model knows the right answer. And so it can check its predictions against the right answers and do that over and over and over again. And also because this is a part of the transformer architect, it turns out that this training is kind of neat in the fact that the way the math happens, that a lot of this computationally heavy math can be done in parallel so that training can be faster. And then I'll just drop this and not really talk too much about it, but it has attention layers. There's a paper out there that's pretty seminal called Attention is All You Need. And so for these BERT models, these attention layers are super important. And just as a kind of broad overview, what these self-attention layers do is for every layer in the neural net, there's another layer. It's not really a layer. There's another piece of data that matches all the inputs for a particular layer. And it gives a weight for each of those layers so that at any point in time, this attention layer or attention mask will tell the model what pieces of this layer, this neural net layer, is important for whatever it is it's trying to do. So it's not just a bunch of words, and actually it never is words, it's really numbers, but then this extra attention layer kind of lets it tweak and pay more attention. Like, should I pay attention to grammar? Or should I pay attention to context? Or, you know, if this is a verb, is it past tense or present tense? So it's really pretty cool. So now that I talked a bit about BERT, so again, we started with the large language model, then we talked in particular about BERT, bidirectional. And now I want to talk about two other models, and they are BERT models, but they're specific to proteins. So you may remember the first thing we talked about was drugs and dealing with proteins to try to create drugs. So this is finally, we're circling back around to drugs. So there were two models that I played with a lot in my previous position. One was called ProtBERT, which was developed by the Ross Lab. They're located in Germany, in Munich. And another model called ESM2, and that was developed by Meta, Facebook. And ESM stands for Evolutionary Scale Modeling. The reason this is kind of interesting is the way they trained this model is it captures what we think of as evolutionary relationships. Evolutionary relationships. So proteins do kind of mutate over time. And so ESM kind of tries to look at that evolution over time. So again, this is a large language model, because in this case, instead of looking at words, we're going to use amino acids. We're going to think of amino acids as a word. And the whole protein will be like a sentence. So essentially, if I have a protein and I mask out 15% of those amino acids that make up a protein, I want it to predict what those masked out amino acids were. And I'll show you some pictures in a minute, actually in the next slide. But the thing I want to stress here is that both these models, ProtBERT and ESM2, they're not that BERT model I talked about a minute ago and then fine-tuned. It's not that at all. Really, it's trained from scratch. There are tons of databases out there for free that has a lot of information about proteins. It'll name a protein, it'll tell you all the amino acids that go into making up the protein, and it'll have certain characteristics like, I don't know, binding or metabolism numbers. And so in this case, this is not the earlier BERT model that was fine-tuned. This was trained from scratch from all these proteins. And again, someone was kind enough to just put these models out there in Hugging Face for me to walk by and pick them up and download them and let me play with them. So maybe I should have done this earlier, but I'll just do a little overview about proteins and amino acids. I probably should have done this earlier as I've been talking about proteins, but so you may remember from high school, proteins are considered to be the building blocks of life. They're very large, they're pretty large and very complex molecules. And they're needed for everything in our body, for tissues and organs. But these proteins are made up of amino acids. And so a lot of people often talk about the proteins look almost like a string of pearls, where each pearl is an amino acid. So an amino acid is the building blocks of proteins, and there are 20 amino acids. So what's interesting about this is you can think of it almost like there are 26 letters in our alphabet. Well, there are 26 amino acids in the protein alphabet, if you will. But in this case, they're not really an alphabet that makes up a word. They're actually more like a word that makes up a string, a sentence. If hopefully that I didn't just confuse you. So what's really interesting about these string of amino acids, which are proteins, is once you string all these amino acids together like this, like pearls, then they end up folding on themselves. And it's this folding that is really important. So when the protein is strung out straight, it's not very useful. It's only once it starts to fold. And so some of the common shapes it folds into is a helix here, or these sheets here. And this is called a secondary structure. And then what happens is these structures, these sheets and helices, they then combine even further to make really complex shapes. And so this is what we're talking about when we're talking about protein. And so to give you an idea of, if I change this, actually, I think I have an example next. And then then this shape then will meet up with a bunch of other shapes. So there are layers on layers on layers. And so this is incredibly, incredibly complex. So let me give you an example of what I'm talking about with if there's one amino acid that changes. So let's just look at a red blood cell. So a normal hemoglobin red blood cell. So normal, or is typically called wild type in protein space. They just call it wild type, meaning typical. So a typical healthy red blood cell has a bunch of hemoglobins that are sort of globular, and the whole shape is disc shaped, and it's kind of got this indentation. But what's important about this is this shape makes it so that these red blood cells can flow through blood vessels pretty easily. And so it turns out that if you change just one amino acid, if we just change glutamate, that E, it's actually an E, to a valine, a V, now your red blood cell is sickle shaped. And now this person has sickle cell anemia. And so when the blood cell changes from this nice disc that flows freely, changing glutamate to valine, now this blood cell is sickle shaped and it does not flow through the blood vessels well at all. It clumps up, it forms blockages. It's a bad deal. So, and that's just with one amino acid changing from glutamate to valine. So my goal is to try to find, if I mutate one of these amino acids, what happens? Is it a good thing? Is it a bad thing? Does it not make a difference at all? And so these models, the ProtBERT and ESM2, try to predict what happens if, for example, I mutate, programmatically mutate a glutamate to a valine. Okay, so now back to this again. So remember what I was talking about, this is what I hope to do, is this in silico screening. And so again, back to this deep mutational scanning mat. as I change each one of these letters in this protein, when I mutate each one of these amino acids to a different amino acid, and I can mutate it to 20 different things. Now I shove this data through this ProtBERT or this ESM2 model, and it will predict something for me. So again, here's the input where I say, all right, instead of E. coli, I'm going to mutate one amino acid, push that through this model, fine tune it with my data, and then it'll give me a prediction of data that I'm interested in. I'll just pause here for a second. Do you have any questions? That's probably a lot. Yes, please.
How does that happen? Like, what kind of parameters is it taken into consideration? Because it's not just linear relationships, right? You have entangled correlations.
But that's the thing, it's not linear. That's because we're, and it's so not linear, because if you even think about the protein, about how it folds and joins, it's an incredibly crazy relationship in the fact that there are some of these amino acids that have like, they're polar, they have a charge. So two positive amino acids don't want to be next to each other, they repel each other. And that's not linear at all, because some of these amino acids have that trait and some don't. And then there's some other amino acids that just really are attractive for other reasons to other amino acids. So when things fold, it will more naturally try to match up those amino acids that pair well. So for example, if I change one of the amino acids from being a positive to a negative charged amino acid, it could be that that protein just will no longer fold, because as the fold starts to happen, and let's just say these two amino acids now repel, so it starts to fold, but they repel, and then maybe the amino acid, the protein won't fold at all. So that's not going to work. That's not going to create a drug at all, because the protein's not folding. It just failed.
And we have models. Yeah, no, it does make sense. It's very interesting. And we do have models so far of what's going to happen when you change one or two or more amino acids, right?
So yeah, so but we don't know it for everything. That's the thing, is we don't know it for every protein and every mutation. We only have a very small amount of information based on what could be. So in this, let's just pretend my amino acid down here is like 100, 200, my protein. Let's say my protein down here on the bottom is 200 amino acids. There is not a database that went through and said, all right, I'm going to change each individual amino acid from its wild type value to every single one of these 20 values. Because actually, I mean, that's what people do in the lab, is they take these plates with wells and they squirt stuff in for each well, and it's really tedious, it's error prone, it can be inconsistent. So people don't do it for everything. They only do it for things that they think are promising. So that's where I step in, is I try to lead my chemists and I say, I think this is promising, and I think these are not promising. And in fact, just before I left, we ended up creating an ensemble model, of which I contributed 2 models. We were able to identify, I want to say something like 12% of the samples as being interesting, whereas the chemists, when they were doing it by hand, they didn't see them at all. So it was really, and then once we said to them, we think these are promising, the chemists would look at it and say, oh yeah, I can see why this is promising, because, you know, the second carbon, blah, 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 blah. You know, I don't understand that. I can just run these models. But when they looked at it, they're like, yeah, actually, this makes sense. Thank you.
May I ask a question too?
Yes, please.
So how do you evaluate like interesting and or bad or good, right? I think that's really difficult.
Yes. And so one thing I don't think I went into in great depth with this picture here, but let me try to do that, is there is an there is a protein, a string of amino acids that I know a certain value. Let's say it's binding strength. So let's say I want things to bind strongly. So let's say this is E. coli. And so we do know the binding strength of the protein E. coli. We know what that binding strength is. And someone has sometimes said, well, if I change one of these amino acids in this protein string and measure the binding of that, I get a new value. And so people have done that a little bit. And so maybe I have that data for not E. coli, but other proteins. And it turns out lots of proteins are very related to each other, or certain sections of the protein can look a lot like another protein. And so essentially, just like these large language models where I say, I went to the blank to make a deposit, that blank thing, I don't know, let's pretend nobody has ever written or the sentence, I went to the bank to make a deposit. Obviously someone has, but let's pretend someone hasn't. But through these large language models, it's able to make that guess because there's enough similarities and there's a whole cloud of words, or in this case, a whole cloud of amino acids that makes sense when it is plugged in that sentence, or in our case, makes sense when that amino acid is plugged into this protein. Because again, some amino acids repel, some come together more strongly. And so just by having a bunch of libraries of amino acids that make up proteins and their characteristics, I can do this fine tuning for my data. Let's say I care about binding strength. It's just, to me, it's like magic. And I know I shouldn't say that because I'm the person who's always annoyed when people think data science is magic. But I got to tell you, these large language models feel like magic to me. I don't know if I ever answered your question.
So one more question, sorry.
Yes,
So
please.
if you go back to the picture, this graph, what does these values represent?
Yeah, good question. I think I talk about it more later, but now is a good time to address it. No, now is a good time to address it. So again, because this is data from my previous company, it's proprietary. So that's why I masked out what this actual protein is. It's not E. coli. It's a drug that we're trying to develop. But let's just put, so I'm just going to call it E. coli. That's a lie, but it's E. coli. And let's pretend that this y-axis, not the y-axis are the mutations. Let's pretend this color represents some measurement of this protein and say the measurement is binding strength.
Okay, got it.
So, but I will say that what this color represents is a relative binding strength. So 0 is the binding strength of the original protein before I started to mutate each amino acid one by one. So if it, let's say it is binding strength, maybe I want binding strength to go higher. So anything above 0 is interesting. Hey, check out these 2 red spots here. That's pretty amazing. And again, if it is binding strength, and if the relative binding strength after I mutate a single amino acid, if it decreases and it starts to go blue, binding strength is weaker. It's probably going to not create a drug. And so I'm going to tell my chemists, all of these blue mutations, don't bother. Hey, check out these 2 bright reds. And hey, look at this patch. Whatever these amino acids are here, if you mutate them, there are a lot of different mutations where all of a sudden you're seeing that this binding strength, which it's not binding strength, that's why I'm using air quotes, look, it increases. And again, like when I showed them these bright red spots, they're like, oh yeah, the benzene ring and blah, blah, blah. I can see why this happened. And I was like, okay, cool. So does that help?
Yeah, so I can, so just reiterate. So you take a feature, and that feature of the whatever you're looking at, I don't know how you define the feature, but I would
binding strength. Let's call it binding strength.
So let's say this binding, and then through I don't know, training, you look at the final numbers, and whenever you plot that, then from the plot you can figure out what are the favorable changes to
Yeah.
you. That's what you present. Okay, perfect.
Yeah, yeah.
Hey,
Because-
Betty, I have a question.
Yes, please.
Thank you for the presentation. It was super helpful. So I have some basic question where, so you're trying to use a model that's a combination of BERT and ProtBERT.
So
Um,
actually, it's only ProtBERT. It's only ProtBERT.
Okay,
ProtBERT is built on, I'm sorry, ProtBERT is like a BERT, but it isn't BERT. So BERT was trained on English language.
mhm,
ProtBERT was trained on a database, many databases of proteins.
okay. And also, when you say you're doing a model training, is it some kind of language that you use which supports a certain, like Python or R?
Right,
Um,
I am using Python,
mhm,
and I'm using PyTorch, and I'm using a bunch of code that's just out there in Hugging Face.
Okay,
So it's pretty amazing. So I'll also mention that I'm like not even halfway through this talk, but I think I will not go into the training part, and just because I think we only have like 6 minutes left. So keep asking questions, and maybe some other time, maybe I'll complete the rest of the talk. But in case this is helpful, I'm just going to leave this up, because I think this is the most interesting way to talk about this. So are there any other questions? Oh, but you know what, actually, you know what else might be helpful? Let me see if I can bring this up. So this is how the training happens, actually. So you remember these large language models that I was talking about. So you can think of this as a large protein model. But the input, instead of being I saw a cat, instead it's a sequence of amino acids, where it could be E. coli, where I've mutated a single amino acid in here. I pass that through ProtBERT or ESM2, and then there's this output. But what I do is I interrupt that output and I push it through one more layer, where what goes into this layer is the output from ProtBERT or ESM2 plus my data that measures. So I can say for some amino acids some proteins that I know of, I know their binding strength, let's say. And that's the additional data that goes into this last layer, my data, as
Right.
well as the output from ProtBERT or ESM2.
Oh, okay. So how do you collect that data? Is it like a database that exists already?
So in this case, for me, I trained this on data I found out there on the internet. And then I had it predict on data that our chemists developed. So the model never saw the in-house data from my chemists.
Mhm.
So I trained it on data that I found for free out on the internet, trained it with this fine tuning. And let's say it predicted binding strength. And then I took my own binding strength data from my chemists. And I said, all right, now that I've fine-tuned this ProtBERT or ESM2 model, go ahead, I'm going to give you data you've never seen that my chemists created, and you go ahead and predict what the binding strength is.
Thank you so much.
Sure. Any other questions?
So I have a curiosity about, so you keep talking about binding strength, and I know that's just an example, but it seems like if binding strength goes up and side effects also go up, when are you bringing those outputs together and making some choices there? Does that make any sense?
Yes, absolutely. And I'm glad you asked that. So the way I'm presenting this is overly simplistic. So
Right, that's what I was thinking.
Here's my binding strength. But this could also be binding strength and toxicity and rate of metabolism. So I could have a lot of data that I stuff through this fine-tuning layer. It isn't always just one piece of data. It could be many pieces of data. And the other thing that's interesting about that is I could also do like a chaining of well, maybe I only train it on binding strength. And when I see things of interest with more binding strength, maybe I pass it through another model that predicts toxicity. And so I could take these proteins that my first model identified as of interest because the binding energy increases in comparison to wild type. Then I could take those and pass it through my second model that measures toxicity. And maybe of all those that I see with increased binding, maybe 10% are less toxic, which is what I want. And so we can do this over and over and over again, is I can take a protein that my chemists are interested in, and I can throw it through multiple models and try to, there's this funnel where I'm trying to direct them away from things that aren't going to work or that are more toxic or have less binding, and toward things that are better for our drugs, for drug development.
Right. Thank you. I guess, and I'll push this a little further. You talked about changing one amino acid. And I'm assuming that's also a simplification. You change one and then, or no, is it always just one, or you're changing and then that's chaining as well? Is that correct?
Right. So you're right, it is simplistic, but I will say that's all I've ever done because it's super hard to find data where people have changed more than one amino acid, because then the permutation is so huge.
Right.
That you just can't even fit it into, memory or anything like that. So the only data I've used and the only data I've seen out there on the internet is a flipping of one amino acid. But you're exactly right. That is overly simplistic. That really isn't enough. And that's why chemists will never lose their job, because they have a better intuition of this. But because the landscape of what they could investigate is so huge, even my overly simplistic work is a huge benefit to them, because they can all of a sudden ignore like 75% of the space. I made up 75%.
Right, thank you.
So I know we're right at time, but I'm happy to hang out. If you need to drop, please do, or we can just end it right here, and I'm happy with that. Last call, any questions? All right, well, thank you for this opportunity. And if you do have questions, feel free to reach out to me outside of this if you wish.
We should at least give a hand.
Thank you
Thanks
for
so much,
presenting that.
BettyAnn. Thanks so
Thank
much.
you. Thank
Beautiful
you so
talk.
much.
You're welcome. Thank you.
Thank you.
Thank you.